thank you everyone for joining us. We are BrainXell, and we are here today with Gently Soto Albrecht with CureC9, and we would love to talk about this new exciting partnership that BrainXell has embarked upon with Gently. I'm going to hand it off to Mike Hendrickson. He is our VP of Operations here at BrainXell to talk through the science and what we're doing in this partnership. So Mike, take it away.
I'll start out gently with a question for you about C9orf72 specifically, which of course is the most common genetic cause of ALS and FTD, and which also makes it an important general cause of those disorders. However, we know that research in this area has been really challenging, and maybe you could speak to that a little bit from your perspective. As why is this still a major issue for the research community?
Thank you so much for asking that question, Mike and Addie, for the introduction. And I just want to start by saying BrainXell is one of the first organizations to ever partner with me in my attempt to search for a way to accelerate a cure in my lifetime. I'm a genetic carrier for C9orf72, that gene that Mike introduced, and it's a repeat expansion. And actually, it's one of the reasons why it was so difficult to discover this mutation, because the same approach that we use to detect it is the reason why it expands, and that's that the DNA polymerase gets slippery and can slip off, and so it makes it really difficult to detect the mutation. And so it was detected in 2011, which is pretty recent, and it was found to bring together both amyotrophic lateral sclerosis, or ALS, and frontotemporal dementia, or FTD. And before then, the two diseases were not known to be associated. But since then, the models to be able to develop therapeutics and better understand the biology of C9orf72 have been difficult. So normally we think about disease models to be cell culture systems and mice. Well, it turns out that the mice that we have don't both show a good pathology and disease progression, and certainly oftentimes they don't show both ALS and frontotemporal dementia type symptoms, so behavioral and motor, which you would expect to see in a perfect mouse model of the repeat expansion. And then when it comes to cell types, there are several banks of induced pluripotent stem cells, or just stem cells for short, which are classically used to make motor neurons and other type of neurons to model neurodegenerative diseases. But the C9 ones, first of all, they're kind of scattered. They're mostly from symptomatic patients, but sometimes it's not known if the patient, what disease they had, or whether they were asymptomatic at the time of collection. So the clinical data associated with them isn't very good. And then sometimes the same cell line gotten from the same place has a different pathology. And by pathology, I mean this repeat expansion forms 7 different types of things that clump up in our neurons. 2 different RNA foci, plus and minus sense, as well as 5 different dipeptide repeat proteins, or DPRs. And then there's TDP43 pathology. And a perfect cell model should recapitulate these if it's really recapitulating disease. But in some people, you'll get some of these, and in some other people's hands, you'll get other pathology. And so they're hard to get. They're kind of scattered. And I think probably the thing that I most recognize when I entered this field is they're actually technically difficult to get access to, even if you're in academia, and sometimes near impossible when you're in industry. So for example, in order to get cell lines from an academic biorepository, let's say you're in an academic lab, your institution has a bunch of paperwork that you're trying to get through the other institutions, and the lawyers are talking, and it could take a whole year for you to get access to cell lines, and sometimes it never happens. And sometimes the cell lines are never made available to industry. But industry and startups have the resources to develop treatments for us. So it's really important that they get access to the cell lines too. So just to summarize, because that was a lot, where I wanted to introduce the C9 biology and some roadblocks. The C9 biology is very, so C9orf72 or C9 is very complicated. And so it's very important to model disease in either mouse or cell models, but those are imperfect. And even when they exist, they're hard to get access to. And so our collaboration with BrainXell actually, I truly believe is going to accelerate progress towards developing better C9 cell models with the best type of clinical data associated with them from multiple families, from symptomatic and pre-symptomatic patients, where you can get access to either the stem cells or differentiated cell lines later with just one step before differentiation. And I'm really excited to work with BrainXell to bring this resource to the research community.
So thanks for that answer, Jenny. That was great. So one thing that, you know, as part of our partnership, we plan to do is to make multiple cell types from these stem cell lines, these induced pluripotent stem cell iPSC lines. And so I'd like you, kind of your thoughts and comments about how you and the field are thinking about the importance of not just the motor neurons, which of course are certainly important for these disorders, but also the astrocytes, microglia, and other cell types, and how the field sort of views these glia that were maybe previously somewhat ignored, I think, by the neuroscience community in general.
That's right, Mike. I mean, I would say generally we don't know how neurons are dying in ALS, certainly in C9, ALS, and frontotemporal dementia. And we don't know if neurons are the most important cell type in the disease. Or certainly we don't know if they're the most important driver of disease. It could be the supporting cells, because neurons need glia to survive. And so if something's wrong with the glia, then the neurons die. And we know the neurons are dying, but we just don't know it's originating from neurons. So it's important to be able to model not just neurons, in addition, motor, but other types of excitatory neurons, because dementia needs to be modeled with a non-motor neuron subtype. But to make the different astrocytes, microglia, and in some cases, maybe in the future, oligodendrocytes, although those are challenging to make. And then things like muscle cells, those may be important, and there may be co-pathology at the motor end plate, the neuromuscular junction. So it's important to be able to make all of these from a C9 ORF72 repeat expansion stem cell, but to make high quality versions that are easily available, where you just are purchasing something that's been validated in a vial without needing to go through a whole year of signing paperwork. And it's important to have biological replicates, so have different, not just have one or two different cell lines available, but have multiple from the same family, from multiple families, so you can see, is the phenotype that I'm seeing tied to one family, or is there a specific other genetic component that's driving the diversity of phenotypes that we're seeing, or the different responses to treatment? We don't know if there's just going to be one treatment for C9 ORF72. That'd be great. It's resources like BrainXell's C9 biorepository that are going to make asking and answering this question possible.
And so how do you see, you know, let's say, obviously we're working together and with other people in the community as well, you know, to make these resources more readily available. And as you said, we've seen this as a bottleneck in the field, getting material into the hands of, especially of industry researchers, but even academics, yep, in a timely manner, right? Because we know that patients,
Mike, it took me 12 months to get to my cells. So like in academia, it happens too, even if they're your own cells.
Yes.
Paperwork.
So, yeah. So how do you see access changing, and this is a difficult question, but changing the way the research is done in the field, hopefully accelerating it, of course, compared to some of the models that have been used, you know, and pre-iPSC models, or models that are still used that maybe are not as relevant?
Right. So the gold standard is definitely to work with a neuron derived from a stem cell, and that's because you can work with, you know, glioblastoma cell lines, which are neuron-like, but cancer cells, which is how a non-stem cell derived neuron is able to survive, they have different biology. So it's really important to model these neurodegenerative diseases in cell backgrounds that come from patients, and you're not going through some kind of arbitrary system to allow the neurons to survive in a dish. But that's not the only way in which it's going to be a game changer. There isn't a resource like this for such an important genetic cause of ALS and FTD, where and I can't stress this enough, you're getting a bunch of different stem cells from different people in different families, pre-symptomatic and symptomatic, and the quality control of the stem cells and the differentiated cell lines that you're getting is industry standard. So you're not working with a biorepository in academia that was made over the course of 30 years by different people using different protocols. It is all done by the same protocol, oftentimes the same people within a company, and you're testing quality control metrics to make sure that your product is up to snuff. And then you're selling them industry competitive prices so that it's not cost prohibitive to get access to these cell lines. And I think that's going to be a game changer in addition to it actually providing a resource to some individuals in industry and academia that have not been able to access even the cell lines that exist. And I also say that there's actually a lot of interest from the patient community. I've had about 10 people reach out to me, C9 carriers, that want to give their cells, their biopsies. Actually, my cells and my dad's cells are the first ones seeding this biorepository, and it was like a little piece of my skin that was removed. And they just put a Band-Aid, it like bled a little bit, and then it was fine. And it's like pretty simple. And there's so many patients now that want to contribute in that way. And I think one of the most incredible things is that we've actually negotiated that a percentage of the profit is going to come to the cost of buying these stem cells is going to come to End the Legacy, which is the organization supporting genetic carriers predisposed to ALS and frontotemporal dementia. So some of the proceeds are actually coming back to the patient community, in addition to us using our bodies to remove barriers for other people to generate therapeutics to maybe give us a chance to live.
Yeah, I think that's a great aspect of it's sort of this, partnership between patients, nonprofit organization, and BrainXell, which is a commercial entity, of course, and then our customers who may be in academia and or in industry, correct?
I've had people ask me, why don't you just develop stem cells and animal models of your own cells and your dad's to see if you can cure yourself? And I tell them, I've got one better for you. I don't just want to cure myself. I want to accelerate a cure for everyone with C9 and for a lot of other type of neurodegenerative diseases, not just ALS and FTD. So I want to create a resource where scientists are able to figure out if their therapies work in my cells and my dad's cells, but also if their therapies are broadly applicable to all C9 or if there's only specific subtypes that they work in. And it's really important to me, based on my medical training, to advance treatments for all patients, not just myself. And I kind of like the look of surprise that people have, because what use is just curing yourself when it's important to advance treatments for like the entire disease. And that to me is just a very easy kind of way to think about it. But that brings me to the fact that my cells and my dad's cells are the seeding cell lines of the BrainXell CureC9 biorepository, and they're currently being made into stem cells. And then once they've passed the stem cell quality control, we are hoping to add about one cell line a month through a collaboration we have with another entity to be disclosed later. And those cell lines are going to have a lot of really important clinical data and family history and a subset of whole genome sequencing associated with them, as well as maybe the opportunity to request PBMCs or other immune and blood cells correlating with those patient cell lines. So this is going to be an unparalleled commercial resource. And all us patients ask for is that the individuals or companies or organizations that are buying these cells use them to also accelerate a cure or really effective treatment for us.
That's great. And yeah, you're just kind of leading into it, but I hear it in your voice, and I hear it in the data you're bringing to this conversation. But what gives you hope as you move forward in progressing to find cures for this disease? What gives you hope in the future of ALS and FTD research?
I find a lot of hope in action. For me, the hope is encompassed by both a global program called CureC9 that I started, Cure, as in finding a cure, and then C9, as in C9orf72. You can read about it at CureC9.com, where we also, under projects, have a tab for the BrainXell C9 biorepository. So that's where all my collaborative projects live, including the ones where I'm either making resources for the research community, like this one, or I'm reducing barriers for the research community to continue working, including work to consolidate all data that exists on C9, and I'm working with some different people to do that. So a couple of individual projects, and then on the community-facing aspect, I have Search for a Self-Cure video series bringing the science to patients. So I think it's really important to do this. The CureC9 research program, that's kind of within the CureC9 umbrella program, is a research. It's a 501c3 under Everything ALS, and that the mission of CureC9 is to meaningfully alter the course of C9 disease, which right now is ALS and FTD, both of which are completely fatal. And I have, to that end, we have an amazing scientific advisory board, which you can read about, people that personally inspire me, people that have played a role in other gene therapy approaches to C9 who are currently developing novel ones. And if you want to support financially, get involved or volunteer with the organization, or if you're a patient and you want to contribute skin cells for us to make stem cells from you, you can find all this information on the CureC9.com website, and there's ways in under every project to easily fill in who you are and how you want to participate, or you can just go to the contact site and do it there. So yeah, there's many ways to be involved, but that's what hope looks like to me on a daily basis. I'm fighting with my patient advocacy, with science. I have a PhD in molecular biology, and then with medicine, I'll finish my doctorate in medicine in December from the University of Pennsylvania. So that's trifecta, and as part of that, these partnerships, such as the one with BrainXell, are very, very critical to advancing our mission to CureC9.
Wonderful. Thank you for that. Yeah, it's truly unique to have all of these dots connected: the patient, the researcher, the 501c3. Everybody can play a part in moving this hope forward. So thank you for bringing this to light. And I just have one more question. You've touched on it a bit, but if you could expand, if you had one message to share with researchers about the work you're doing and the field, what would it be?
Remember what your ultimate goal is. And your ultimate goal as a scientist in this space is not to publish. Your ultimate goal isn't to prepare for that lab meeting or presentation at an outside conference. It's not to get another job or to rise in a position in your field. If you're working in the ALS or FTD or C9 space, and you're working on a translational project, your goal is to accelerate treatments for patients. So sometimes that means making decisions where the goal is the winner, and you are setting aside kind of your own desires to advance yourself. And that's something that I've learned as a scientist. The way I approach helping others succeed in this space, lowering barriers for other scientists to advance treatments or biomarkers, connecting people without asking for anything in return. When you're working on a translational project and every patient with these diseases has died and will die unless there's a treatment, and there's thousands of people with that genetic mutation facing these fatal diseases with nothing to help them, then keep your eye on the ball. And the ball is meaningfully altering C9. So you can join us at CureC9.
That's wonderful. Thank you, Gently. Thank you for putting the human element into the science And on behalf of BrainXell, we're just so thankful for your hope and your expertise and for sharing your story. We really appreciate it.
Thank you so much, Addie and Mike. And I really can't stress this enough. BrainXell was one of the first organizations to work with me. Your team has been really patient-centered from day one. How can we get patients' cells to the people that have the resources to advance cures? How can we help patients fight with their bodies? And I just feel privileged to participate in a team effort to allow other people to join the fight. So thank you.